Hello everyone and welcome to another edition of Zog Science. Today we're going to be talking about different patterns of evolution. So the rate of evolution, alright? Scientists use two basic terms to kind of describe the rate of evolution, and the rate of evolution is how quickly is evolution occurring. We've got punctuated equilibrium, and this describes patterns of long, stable periods interrupted by brief periods of more rapid change. So what we can think about if we want to look at this visually, okay, if time is on our y-axis and morphology or kind of how evolution is occurring is on the x-axis, we've got time occurring, right? Time's occurring, and then we have a brief period of very large amounts of change, and then nothing's really changing, and large amounts of change, nothing's really changing, large amounts of change, so on and so forth. Okay, so it's kind of the idea that evolution occurs in short, quick bursts, and a lot of evolution occurs during that time span. We also have gradualism, and gradualism describes a slow, steady change in a population over time. So here, instead of it being kind of straight lines, which denote things are not changing or changing very rapidly, over time we slowly kind of branch out. And the thinking right now is that there are both types of evolution that occur. So you've got gradualism, which is just kind of occurring all the time, but if you have some sort of catastrophic event where a species is introduced to an entirely brand new environment, or there's some sort of major environmental change, that punctuated equilibrium will occur as a species has to rapidly adapt to those changing environments. So there are several different ways that evolution can occur. All right, we've got adaptive radiation, which we've mentioned before. That's where one species gives rise to many species that appear different externally but are similar internally, right? And that kind of makes sense because they started off as a single organism, but they have over time evolved into many organisms. So yes, they might fill different niches. They might eat different things, have different appearances, but internally they're going to look similar. That's going to be kind of like we were talking about the homologous structures, okay? This is also called divergent evolution or speciation. Speciation just saying that we are creating new species. So when does adaptive radiation occur? It usually occurs when there's an empty niche in a community following an event or mass extinction, right? So we've got some sort of mass extinction, a bunch of species die. Now one species is going to begin evolving to fill a lot of those different niches. Remember, a niche is where an organism lives and its role in the environment, right? So it's habitat and what it actually does in terms of, what it eats, what it is eaten by, all those kinds of things. No two species can have the same niche. This is called the competitive exclusion principle. So species compete for the food and resources within a niche, right? So the most competitive species or the most fit species for that niche is going to survive and maintain it. The less competitive species either dies out or finds a new niche, right? So you've got competition occurring between species for resources, and that's what helps drive evolution. Speciation is the development of a new species. Kind of mentioned that before. It occurs when a species moves into an unoccupied niche, right? It's going to be changing into that new species. Over time, the species evolves adaptations that are more suitable to its niche. So adaptations, you can think of them as being like traits, okay? It's just kind of another word for traits. And they're just occur based on the whatever niche you particularly have. So for example, if you live in trees, right, a good adaptation would be to grow claws that allow you to grasp onto the tree so that way you don't fall off, right? Those claws are not going to be particularly useful if you're just walking around on the ground, which is part of the reason why you see birds being awkward when they're on the ground, because they're not adapted for that environment as much. So when does speciation occur? All right, speciation can only occur when populations are reproductively isolated from one another, right? And notice that we're saying that they have to be reproductively isolated. They don't have to be geographically isolated necessarily, because speciation just occurs when two species are no longer mixing their alleles, mixing their genes with one another, right? Their gene pools with one another, so therefore they each are evolving independently, okay? So reproductive isolation means the population can no longer interbreed with a population in other areas. So remember, we talked about this kind of before. You can think about the gene pools, right, if they're connected to one another, that means that there's breeding going on between them, so alleles are being mixed, all right? But if they get cut off, right, if those individuals that are going back and forth between the populations die off, or there's some sort of event that stops that, now they're isolated and they evolve independently, okay? So again, it only happens when the gene pools are separate. There are kind of three basic factors that maintain reproductive isolation. We can have geographic barriers such as mountains, rivers, streams, oceans, whatever. Okay, different courtship behaviors, right? So if you've seen different birds, right, they all have different kind of songs, right? So they're different songs. Other birds don't hear that. They don't interpret that as a courtship, so they're not going to be attempting to mate with that species. And then also physical differences, right? Looking different, anatomy differences, all that kind of thing. So geographic barriers, we've got a beetle population, right? There's some sort of river that arises. It separates it into two different populations, and now when the river dries up, they cannot actually breed with one another because they have been isolated. Again, we can have all kinds of different things that can cause those geographic barriers. Courtship behaviors, differences in mating rituals or seasons. Okay, so different plant species pollinate at different times of the year. Therefore, even though they might live in the same region, their genes are never going to be mixing. And then physical differences in size, shape, all that kind of thing. So, speciation via reproductive isolation. A reproductive isolation occurs between two populations of the same species. This leads to the populations to become adapting to different environments, right? Different environments, which means that they are going to evolve different adaptations. The separate gene pools become less similar. Eventually, reproductive isolation becomes permanent and two different species form, right? So it kind of progresses on through that. So here's our finches that are found on the Galapagos Islands, right? Notice that they all have different beaks, right? They have different colorations. Again, that is because they have been evolving to different environments. Speciation doesn't necessarily always happen. With the gene pool for populations very small, there is less variation. This makes it difficult for populations to adapt to a new niche. So if you have a small population, they might not necessarily evolve into a new niche. And these populations often become extinct. So a lot of the endangered animals that are in the world, they have a very small population, which means that they don't have a lot of genetic variation, so they're more susceptible to things like diseases and other catastrophic events. We also can have convergent evolution, so we just kind of talked about speciation producing different species. We can also have different groups developing similar adaptations because they live in the same type of environment. So a great example of this is the ocean, right? So we've got dolphins, sharks, and penguins, right? They all kind of live in the ocean, or at least spend a majority of their time in the ocean. In the case of penguins, they do a lot of swimming in there, hunting, that kind of thing. So they all have developed kind of very similar body characteristics, right? A very streamlined body. They have fins, right? The wings of the penguin act like their fins. So that would be convergent evolution. An example of that would be analogous structures from our evidence for evolution, right? They live in the same environment. They have very similar body plans. We can also have coevolution, okay? Here's where two unrelated species become uniquely adapted to one another's, right? So for example, we talked about this before, mitochondria and the eukaryotic cell, also chloroplasts, right? They started off as separate bacteria, but then they began to coexist with one another and provide each other with benefits. And we also have plants and their pollinators, okay? A lot of plants only have a single species that pollinates them, and that is what is the only way that they can actually have the pollen transferred from one to one another. This is kind of a big deal right now because of the whole bee colony collapse disorder, where a lot of bee populations are kind of falling, and that's an issue because they pollinate a lot of different flowers, and a lot of agriculture actually relies on bees as well. There's the endosymbiotic theory, which again we discussed when we did our cell unit. So those are our different types of evolution. How do we know that evolution is actually occurring? Okay. Well, Hardy and Weinberg devised a system for determining whether or not evolution is occurring. In order for evolution to occur, allele frequencies must change. Right? That makes sense. Our genes have to be changing. We have to be having new traits. Therefore, our allele frequencies must change. And when they are not, a population is said to be in genetic equilibrium. So when the alleles are not changing, there's no evolution, and we are in genetic equilibrium. Now, this is not necessarily something that's occurring all the time, right? So what we have is we have a set of rules to figure out if a population is in genetic equilibrium. Okay, first of all, we can't have any mutations. Okay, so no new alleles are being introduced. No gene flow can occur, i.e., no migration. So individuals cannot be moving in and out of a population. Random mating must occur, right? Therefore, you can't have individuals choosing other individuals, purposefully. It just has to be kind of random. The population must be large so that no genetic drift or random chance can cause the allele frequencies to change, right? So if a group of a population gets cut off, right, it's not going to change the allele frequencies. And no selection can occur so that certain alleles are not selected for or against. Okay, now, is this ever really actually happening? No, but what we can do is we can pick out a certain moment in time, and in that moment in time we can say, hey, this population at this particular moment is in genetic equilibrium, right? Because if we just pick a single moment in time, all of these conditions can be met. And when those conditions are met, then we can use these lovely equations or formulas in order to determine the allele frequencies within a population. So we've got two formulas, p squared plus 2pq plus q squared is equal to 1, and p plus q is equal to 1. Now, if you'll notice, p plus q is equal to 1. If you square this, then you get p squared plus 2pq plus q squared is equal to 1, right? This is the perfect square, okay? What do these terms mean, okay? P is the frequency of the dominant allele in the population. Q is the frequency of the recessive allele in the population, okay? So P plus Q has got to be equal to 1 because you're either only going to have dominant or recessive alleles. Now, this system can be expanded to deal with multiple alleles, right? Genes that have multiple alleles. You could go P plus Q plus R, right? P plus Q plus R is equal to 1, and then you would square that, and you would get a much larger set of formulas over here, but you can do that. All right, so for this formula, P squared is the percentage of homozygous dominant alleles, 2pq is your heterozygous alleles, and q squared is your percentage of heterozygous individuals. Now, when you're doing these are all going to be in decimal form, so it's the decimal percentage form. So, for example, p plus q is equal to 1. P would be, let's say, 0.6. Q is 0.4, right? We could square that. Now we know our percentage of homozygous dominant. We could square Q. Now we know our percentage of homozygous recessive. And then we take 2 times P plus Q, and therefore then we get our percentage of heterozygous individuals. All right, and that's all I got for you today. Thank you very much for watching, and I'll see you next time.